Hi class, this is your instructor, Skylar Huff. We're now in Chapter 35. This is plant form. Although a cactus, an orchid, and a hardwood tree all look quite different, they share a similar basic structure with all plants. This is reflected in how plants develop, and how they grow, and how they transport substances throughout their bodies. Here, we will address the question of how a vascular plant is built. We will focus on the cells, tissues, and organs that compose the adult plant body. The basic plant body of a plant, above ground shoots and below ground roots, is established during embryogenesis. And growth of an adult plant only occurs in very specific regions. Henceforth, in this chapter, we will explore how a relatively simple set of tissues can be used to produce the great variety of forms we see in adult plants. As you learned a bit before, or will learn later, I guess I should say, the plant kingdom has great diversity, not only among its many phyla, but even within species. The earliest vascular plants, many of which are extinct, did not have a clear differentiation of the plant body into specialized organs such as roots and leaves. Among modern vascular plants, the presence of these organs reflects increasing specialization, particularly in relation to the demands of a terrestrial existence. Obtaining water, for example, is a major challenge, and roots are adapted for water absorption from the soil. Leaves, roots, branches, and flowers all exhibit variations in size and number from plant to plant. Development of the form and structure of these parts may be precisely controlled, but some aspects of leaf, stem, and root development are quite flexible. This chapter emphasizes the unifying aspects of plant form using the flowering plants as a model. A vascular plant consists of a root system and a shoot system. I'll show you that here. Roots and shoots grow at their tips, which are called apices, and I'll call it an apex. The root system anchors the plant and penetrates the soil, from which it absorbs water and ions crucial for the plant's nutrition. Root systems are often extensive, and growing roots can exert great force to move soil material as they elongate and expand. The shoot system consists of the stems and leaves. Stems serve as a scaffold for positioning the leaves, the principal sites of photosynthesis. The arrangement, size, and other features of leaves are critically important in the plant's ability to absorb light and exchange gases. Flowers, other reproductive organs, and ultimately fruits and seeds are also formed on the shoot. The repeating unit of vegetative shoot consists of the internode, the node, leaf, and axillary bud. You can see all of those here, but not reproductive structures. An axillary bud is a lateral shoot apex that allows the plant to branch or replace the main shoot if it is damaged or eaten by a herbivore. A vegetative axillary bud has the capacity to replace the primary shoot. When the plant has shifted to the reproductive phase of development, these axillary buds may produce flowers or floral shoots. Roots, shoots, and leaves all contain three basic types of tissues: dermal, ground, and vascular. Because each of these tissues extends through the root and shoot systems, they are called tissue systems. Dermal tissue, primarily epidermis, is 1 cell layer thick in most plants, and it forms an outer protective covering for the plant. Ground tissue cells function in storage, photosynthesis, and secretion in addition to forming fibers that support and protect plants. Vascular tissue conducts fluids and dissolved substances throughout the plant body. Each of these tissues and their many functions are described in more detail in just a few. Plant cell types can be distinguished by the size of their vacuoles, whether they are living or not at maturity, and by the thickness of their cellulose cell walls, a distinguishing feature of plant cells. Some cells have only a primary cell wall of cellulose synthesized by the protoplast near the cell membrane. Microtubules align within the cell just below the cell membrane and determine the origination of the cellulose fibers. Cells that support the plant body have more heavily reinforced cell walls with multiple layers of cellulose and other strengthening molecules, including lignin and pectin. Cellulose layers are laid down at angles to adjacent layers, which enhances the strength of the cell wall. When a seed sprouts, only a tiny portion of the adult plant exists. Although embryo cells can undergo division and differentiation to form many cell types, the fate of most adult cells is more restricted. Further development of the plant body depends on activities of meristems, found in shoot and root apices, or the shoot apex and the root apex, as well as in other parts of the plant. Meristems are clusters of small, undifferentiated cells with dense cytoplasm and proportionately large nuclei. When these cells divide, they give rise to two kinds of cells, one that replaces the meristematic cell and another that can differentiate into different tissues of the plant body. This maintains the population of meristematic cells while providing a new plant tissue. This is similar to how stem cells function in animals. Reproduction of the stem cell and the production of new cells of different types. Although the molecular program for stem cells in plants and animals does not appear to be the same, there is one protein critical to the maintenance of these cell populations in both plants and animals. The retinoblastoma protein in animals and the homologous retinoblastoma-related proteins in plants both function to maintain the size of stem cell populations to fit the needs of the organism. In plants, the extension of both root and shoot is due to repeated cell divisions and subsequent elongation of cells produced by the apical meristems. In woody plants, lateral meristems produce an increase in the diameter of roots and shoots. Apical meristems are located at the tips of roots and shoots, shown to you here, and shown to you there. During periods of growth, the cells of apical meristems divide and continually add more cells at the tips. Tissues derived from apical meristems are called primary tissues, and the extension of the root and stem forms what is known as the primary plant body. The primary plant body consists of the young, soft shoots and roots of a tree or shrub, or the entire plant body in some plants. Both root and shoot apical meristems are composed of delicate cells that need protection. The root apical meristem is protected by the root cap, the anatomy of which is described a bit later on. Root cap cells are produced by the root meristem and are sloughed off and replaced as a root moves through the soil. In contrast, leaf primordia shelter the growing shoot apical meristem, which is particularly susceptible to desiccation because of its exposure to sun and air. The apical meristem gives rise to three tissue systems by first initiating primary meristems. The three primary meristems are the protoderm, which forms the epidermis, the procambium, which produces the primary vascular tissues, primary xylem for water transport, and primary phloem for nutrient transport, and ground meristem, which differentiates further into ground tissue. In some plants, such as horsetails and corn, intercalary meristems arise in stem internodes, adding to the internode lengths. If you walk through a cornfield on a quiet summer night when the corn is about knee-high, you may hear a soft popping sound. This sound is caused by the rapid growth of the intercalary meristems. Many herbaceous plants exhibit only primary growth, but others also exhibit secondary growth, which may result in a substantial increase of diameter. Secondary growth is accomplished by the lateral meristems, peripheral cylinders of meristematic tissue within the stems and roots that increase the girth or diameter of gymnosperms and most angiosperms. Lateral meristems form from ground tissue that is derived from apical meristems. Monocotyledonous plants called monocots, and I say monocotyledonous plants or monocots are the major exception. Although secondary growth increases girth in many non-woody plants, its effects are most dramatic in woody plants, which have two lateral meristem. One lateral meristem, the cork cambium, is located within the bark of the plants with woody stems and produces the outer bark of trees. A second lateral meristem, the vascular cambium, is located just beneath the bark and produces the secondary vascular tissue. The vascular cambium forms between the xylem and phloem in vascular bundles, adding secondary vascular tissue to both of its sides. Xylem is added to the inside of the vascular cambium, and phloem is added to the outside. Keep that in mind, please. Keep that in mind. Secondary xylem is the main component of wood. Secondary xylem is very close to the outer surface of a woody stem. Removing the bark of a tree damages the phloem and may eventually kill the tree. Tissues formed from lateral meristems, which comprise most of the trunk, branches, and older roots of trees and shrubs, are known as secondary tissues. And are collectively called the secondary plant body. There are three kinds of plant tissues: dermal tissue, ground tissue, and vascular tissue. Each contains highly differentiated cell types, the structures of which correlate with the tissue's functions. Here, the structure and function of plant cells is considered. Dermal tissue is derived from an embryo or apical meristem, forms epidermis. This tissue is 1 cell layer thick in most plants and forms an outer protective covering. The epidermis is covered with an insoluble layer called the cuticle, which functions to limit the movement of water either out of or into the plant. The cuticle is composed of the polyester polymer called cutin and a variety of waxes. In desert plants, several layers of wax may be added to the cuticle to limit water loss. The bark of trees contains dermal tissue, which I'll refer to as the periderm. Epidermal cells, which originate from the protoderm, cover all parts of the plant body. Several specialized cell types occur in the epidermis, including guard cells, trichomes, and root hairs. Guard cells are paired, sausage-shaped cells, and I would even say they may resemble two lips. So they are flanking a stoma, and to be plural, I'll refer to those as being stomata. A mouth-shaped epidermal opening. Guard cells, unlike other epidermal cells, contain chloroplasts. That's a key to learning there. Guard cells, unlike other epidermal cells, contain chloroplasts. Stomata occur in the epidermis of leaves and sometimes on other parts of the plant, such as stems or fruits. Stomata serve a dual function. Exchange of oxygen and carbon dioxide gases through the stomata is essential for photosynthesis. In addition, diffusion of water in vapor form takes place almost exclusively through the stomata, enabling transport of water and minerals from roots to the stems. In many plants, stomata are more numerous on the lower epidermis of the leaf than on the upper, a factor that helps minimize water loss. Some plants have stomata only on the lower epidermis, and a few, such as water lilies, have them only on the upper epidermis to maximize gas exchange. Guard cell formation is a result of an asymmetrical cell division, producing a guard cell and a subsidiary cell that aids in the opening and closing of the stoma. So I don't see much of an easy way to show you the subsidiary cell here, though I can certainly show you all those in lab. Trichomes are single-celled or multicellular hair-like outgrowths of the epidermis. They occur frequently on stems, leaves, and reproductive organs. If a leaf appears fuzzy or woolly, it is probably covered with trichomes that can be seen clearly with a microscope under low magnification. Trichomes are like a Swiss Army knife, you all. Trichomes reduce evaporation by covering stomata openings. They also protect leaves from high-intensity light and ultraviolet radiation, and can buffer against temperature fluctuations, and have most recently been shown to sometimes regulate plant growth and shape by sticking flower parts together to help reproduction. Trichomes can vary greatly in form. Some consist of a single cell, others are multicellular. Some are glandular, secreting a sticky or toxic substance to deter herbivory. These are trichomes, but that is 35 micrometers. Very small. Root hairs, which are tubular extensions of individual epidermal cells, occur in a zone just behind the tips of young, growing roots. Because a root hair is simply an extension of an epidermal cell and not a separate cell, no cross-wall isolates the hair from the rest of the cell. Root cells keep the root in close contact with the surrounding soil particles and greatly increase the root's surface area and efficiency of absorption. As a root grows, the extent of the root hair zone remains roughly constant as root hairs at the older end break off, while new ones are produced at the apex. Most of the absorption of water and minerals occurs through root hairs, especially in herbaceous plants. Root hairs should not be confused with lateral roots, which are multicellular structures and originate from deep within the root. Ground tissue consists primarily of thin-walled parenchyma cells that function in storage, photosynthesis, and secretion. Other ground tissue, composed of parenchyma cells and sclerenchyma cells, provide support and protection. Parenchyma cells are the most common type of plant cell. They have large vacuoles and thin walls and are initially more or less spherical. These cells, which have living protoplasts, push up against one another shortly after they are produced, however, and assume other shapes. Parenchyma cells may live for many years in some plants. For example, in cacti, they may live for over 100 years. They function in the storage of food and water, photosynthesis, and secretion. They are the most abundant cells of primary tissues and may also occur to a much lesser extent in secondary tissues. Most parenchyma cells have only primary cell walls, which are laid down while the cells are still maturing. These are parenchyma cells being shown here, and these are present in a grass plant with only a primary cell wall. If you've ever had celery strings trapped in your teeth, or you see them upon chomping into a petiole of celery, you are familiar with tough, flexible parenchyma cells. Like parenchyma cells have living protoplasts, lack secondary cell walls, and may live for many years. These cells, which are usually a little longer than wide, have cell walls that vary in thickness. Flexible parenchyma cells provide support for plant organs, allowing them to bend without breaking. They often form strands or continuous cylinders beneath the epidermis of stems or leaf petioles, which I mentioned a moment ago, or stalks, and along the veins in leaves. Strands of parenchyma provide much of the support for stems in the primary plant body. These are parenchyma cells. And this is from elderberry. Sclerenchyma cells have tough, thick cell walls. Unlike parenchyma and parenchyma, they are typically dead at maturity. Their secondary cell walls are often impregnated with lignin, a highly branched polymer that makes cell walls more rigid. For example, lignin is an important component of wood. Cell walls containing lignin are said to be lignified. Lignin is common in the walls of plant cells that have a structural or mechanical function. Sclerenchyma is present in two general types, fibers and sclerids. Fibers are long, slender cells that are usually grouped together in strands. Linen, for example, is woven from strands of sclerenchyma fibers that occur in the phloem of flax plants. Sclerids are variable in shape, but often branched. They may occur singly or in groups. They are not elongated, but have many different forms, including that of a star. The gritty texture of a pear is caused by groups, or I'll say clusters, of sclerids that occur throughout the soft flesh of the fruit. Sclerids are also found in hard seed coats, such as the pit of a cherry or even that of a pear. Both of these tough, thick-walled cell types serve to strengthen the tissues in which they occur. And I have never seen a cluster of sclerids that looks like this. Vascular tissue includes two kinds of conducting tissues. One, xylem, which conducts water and dissolved minerals, I'll say upwards, and two, phloem, which conducts a solution of carbohydrates, mainly sucrose, used by plants for food, and I'll say downward. The phloem also transports hormones, amino acids, and other substances that are necessary for plant growth. Xylem and phloem differ in structure as well in function. Xylem, the principal water-conducting tissue of plants, usually contains a combination of vessels, which are continuous tubes formed from dead, hollow cylindrical cells arranged end to end, and tracheids, which are dead cells that taper at the ends, overlap one another. You can see that here. Primary xylem is derived from the procambium produced by the apical meristem. Secondary xylem is formed by the vascular cambium, a lateral meristem. Wood consists of secondary xylem. In some plants, such as gymnosperms, but not flowering plants, tracheids are the only water-conducting cells present. Water passes in an unbroken stream through the xylem from the roots up through the shoot and into the leaves. When water reaches the leaves, much of it diffuses as water vapor into the intercellular spaces and out of the leaves into the surrounding air, mainly through savanna. This diffusion of water vapor from a plant is known as transpiration. In addition to conducting water dissolved minerals and inorganic ions such as nitrates and phosphates through the plant, xylem supports the plant body. Vessel members tend to be shorter and wider than tracheids. When viewed microscopically, they resembled soda cans with both ends removed. A vessel is a stack of vessel members. Both vessel members and tracheids have thick, lignified secondary cell walls with no living protoplasts at maturity. Lignin is produced by the cell and secreted to strengthen the cellulose cell walls before the protoplast dies, leaving only the cell wall. Tracheids contain pits, which are small areas with no secondary cell wall. The primary cell wall is present but is water permeable. In addition to conducting cells, xylem typically includes fibers and parenchyma cells. Some types of fibers likely evolved from tracheids, becoming specialized for strengthening rather than conducting fluid. The parenchyma cells, which are usually produced in horizontal rows called rays, yes, rays exist, and those will be shown in lab, by special ray initials of the vascular cambium, function in lateral conduction and food storage. In cross sections of woody stems and roots, rays can be seen radiating out from the center of the xylem like spokes of a wheel. Fibers are abundant in some woods, such as oak, and the wood is correspondingly dense. The arrangement of these and other kinds of cells in the xylem make it possible to identify most plant genera and many species from their wood alone. Phloem, which is located toward the outer part of roots and stems, is the principal food-conducting tissue in vascular plants. If a plant is girdled by removing a substantial strip of bark down to the vascular cambium around the entire circumference, the plant eventually dies from starvation of the roots. This is also how some bark-grazing animals can cause trees to die. Food conduction in phloem is carried out through two kinds of elongated cells, sieve cells and sieve tube members, or the sieve tube element. Gymnosperms, ferns, and horsetails have only sieve cells. Most angiosperms have sieve tube members or sieve tube elements. Both types of cells have clusters of pores known as sieve areas because the cell walls resemble sieves. Unlike pits in xylem, which contain primary cell walls, phloem pores are completely open. Sieve areas are more abundant on the overlapping ends of the cells and connect the protoplasts of adjoining sieve cells and sieve tube members. Both of these types of cells are living, but most sieve cells and all sieve tube members lack a nucleus at maturity. In sieve tube members, some sieve areas have larger pores that are called sieve plates. Sieve tube members are stacked up end to end in the phloem, forming longitudinal series called sieve tubes. Sieve cells are less specialized than sieve tube members, and the pores in all of their sieve areas are roughly of the same diameter. Sieve tube members are more specialized and presumably more efficient than sieve cells. Each sieve tube member is associated with an adjacent specialized parenchyma cell known as a companion cell. Companion cells apparently carry out some of the metabolic functions needed to maintain the associated sieve tube member. because the sieve tube member lacks a nucleus. So in angiosperms, a common initial cell divides asymmetrically to produce a sieve tube member cell and its companion cell. Companion cells have all the components of normal parenchyma cells, including nuclei. Roots have a simpler pattern of organization and development than stems, so we consider those first. Keep in mind, however, that roots evolved after shoots and are a major innovation for terrestrial living. 4 regions are commonly recognized in developing roots: the root cap, the zone of cell division, the zone of elongation, and the zone of maturation. I'll show you all those here. The boundaries between the last 3 zones are not clearly defined in the plant root. When apical initials divide, daughter cells that end up on the tip end of the root become root cap cells, cells that form in the opposite direction and pass through the other 3 zones before they finish differentiating. As you consider the different zones, think about the tip of the root moving deeper into the soil, actively growing. This counters the static image of a root that diagrams and photos convey. The root cap has no equivalent in stems. It is composed of 2 types of cells: the inner columella cells, they look like columns, and the outer lateral root cap cells, which are continuously replenished by the root apical meristem. In some plants with larger roots, the root cap is quite obvious. Its main function is to protect the delicate tissues behind it as its growth extends the root through mostly abrasive soil particles. The root cap cells, which have an average life of less than a week, are constantly being replaced from the inside, forming muscogels. Golgi bodies in the outer root cap cells secrete and release a slimy substance called mucigel, and that passes through the cell walls to the outside. So this is what eases the root through the soil. The slimy mass also provides a medium for the growth of beneficial nitrogen-fixing bacteria in the roots of plants such as legumes. A new root cap is produced when an existing one is artificially or accidentally removed from the root. The root cap also functions in the perception of gravity. Columella cells are highly specialized, with the endoplasmic reticulum in the periphery and the nucleus located either in the middle or the top of the cell. They lack large vacuoles. Instead, they contain plastids filled with starch grains called ameloplasts. These ameloplasts are affected by gravity and collect on the sides of the cell, facing the pull of gravity. When a potted plant is placed on its side, the ameloplasts drift to the side most affected by gravity, and the roots bend in that direction. The apical meristem is located in the center of the root tip, in the area protected by the root cap. Most of the activity in this zone of cell division takes place toward the edges of the meristem, where the cells divide every 12 to 37 hours, often coordinatedly, reaching a peak division once or twice a day. Most of the cells are essentially cuboidal, with small vacuoles and proportionally large centrally located nuclei. These rapidly dividing cells are daughter cells of the apical meristem. A group of cells in the center of the root apical meristem, termed the quiescent center, divides only very infrequently. So I'm not getting into patterning of this or control of cell division. In the zone of elongation, roots lengthen because their cells are produced by the primary meristems elongate. Because the cells produced by the primary meristems elongate. The small vacuoles merge and grow until they occupy 90% or more of the volume of the cell. The cells that have elongated in the zone of elongation become differentiated into specific cell types in the zone of maturation. The cells of the root surface cylinder mature into epidermal cells, which have very thin cuticle and include both root hair and non-root hair cells. Root hairs greatly increase the surface area and therefore the absorptive capacity of the root. There can be many billions of root hairs on a plant, and they can provide over 37,000 centimeters squared of root surface area. Symbiotic bacteria that fix atmospheric nitrogen into a usable form, usable by legumes, enter the plant via the root hairs and induce the plant to create a nitrogen-fixing nodule around it. Parenchyma cells that are produced by the ground meristem just inside the epidermis. This tissue, called the cortex, may be many cell layers wide and functions in food storage. As described in the inner boundary of the cortex, differentiates into a single layer cylinder of endodermis after an asymmetrical cell division. Endodermal primary cell walls are impregnated with suberin, a fatty tissue that is impervious to water. Yes, it's waterproof. And the suberin is produced in bands called the Casparian strips that surround each adjacent endodermal cell wall perpendicular to the root's surface. These strips block transport between cells. All the tissues inside the endodermis are collectively referred to as the stele. Immediately adjacent to the interior of the endodermis is a cylinder of parenchyma cells known as a pericycle. The pericycle cells divide even after they mature. They can give rise to lateral roots, or in dicots or eudicots, to two lateral meristems, the vascular cambium and choric cambium. You can see what I've been teaching you here. The stele is the section of the root in the center. And that's in both of these, both the eudicot and the monocot. The monocot is at the top. The stele is there kind of with that darkened layer there with the endodermis that surrounds. And then you have at the bottom the eudicot root cross section with the stele there in the center that is quite miniature. And it's endodermis there, or by way of the stele and endodermis. This would be great for studying for your tests. The water-conducting cells of the primary xylem are differentiated as a solid core in the center of young eudicot roots. In a cross-section of a eudicot root, the central core of the primary xylem often is somewhat star-shaped, and I sometimes just call it an X, having from 2 to several radiating arms that point toward the pericycle. And I say it's typical for us to see 4 of those. In monocot and a few eudicot roots, the primary xylem is in discrete vascular bundles arranged in a ring, which surrounds the parenchyma cells called pith at the center of the root. So as you look at this here, please don't confuse this with the cross-section of the stem, which we have not yet gotten to. Primary phloem, composed of cells involved in food conduction, is differentiated in distinct groups of cells adjacent to the xylem in both eudicot and monocot roots. In eudicots and other plants with secondary growth, part of the pericycle and the parenchyma cells between the phloem and xylem become the root vascular cambium, which starts producing secondary xylem to the inside and secondary phloem to the outside. That's a key again, where those are located. And if I were in your shoes, class, I would say, please be sure to see these root cross sections on page, or at least in the chapter, figure 3517. Figure 3517. There's no A or B, though you can see them there. Eventually, the secondary tissues form concentric cylinders. The primary phloem, cortex, and epidermis become crushed and are sloughed off as more secondary tissues are added. In the pericycle of woody plants, the cork cambium contributes to the outer bark. In the case of secondary growth in eudicot roots, eventually everything outside of the stele is lost and replaced with bark. Most plants produce either a tap root system, characterized by a single large root with smaller branch roots, or a fibrous root system, composed of many smaller roots of similar diameter. Some plants, however, have intriguing root modifications with specific functions in addition to those of anchorage and absorption. These have sometimes been referred to as roots modified for unusual functions. Not all plants are produced by pre-existing roots. Not all roots are produced by pre-existing roots. Any root that arises along a stem or in some place other than the root is called an adventitious root. For example, climbing plants such as ivy produce roots from their stems. These can anchor the stems to the tree trunks or to a brick wall. Here we'll investigate functions of modified roots, or those roots that have been modified for unusual functions. First up are prop roots. Some monocots, such as corn, produce thick adventitious roots on the lower parts of the stem. I'll show you all these in lab. These so-called prop roots grow down to the ground and brace the plants against wind. Those are shown here. Adventitious roots are common in wetland plants, allowing them to tolerate wet conditions. Aerial roots. Plants such as epiphytic orchids, which are attached to tree branches and grow unconnected to the ground, but are not parasites, have roots that extend into the air. Some aerial roots have an epidermis that is several cell layers thick, and an adaptation to reduce water loss. Aerial roots may be green and photosynthetic. There are aerial roots. Pneumatophores. Some plants that grow in swamps and other wet places may produce spongy outgrowths called pneumatophores from the underwater roots. The pneumatophores commonly extend several centimeters above water, facilitating oxygen uptake in the roots beneath. These are pneumatophores. And I would imagine some of you all may refer to these as cypress knees. Contractile roots. The roots from bulbs of lilies and from several other plants, such as dandelions, contract by spiraling to pull the plant a little deeper into the soil each year until they reach an area of relatively stable temperature. The roots may contract 1/3 of their original length as they spiral like a corkscrew due to the cellular thickening and constricting. And those are not shown here. Parasitic roots. So the stem of certain plants that lack chlorophyll, such as dodder, produce peg-like roots called haustoria that penetrate the host plants around which they are twined. The haustoria establish contact with the conducting tissues of the host and effectively parasitize their hosts. Dodder not only weakens plants but can also spread disease when it grows and attaches to several plants. Food storage roots. The xylem of branch roots such as sweet potatoes And similar plants produce extra parenchyma cells that store large quantities of carbohydrates. Carrots, beets, parsnips, radishes, and turnips have combinations of stem and root tissues that also function in food storage. Cross sections of these roots reveal multiple rings of secondary growth. Water storage roots. Some members of the pumpkin family, especially those that grow in arid regions, may produce water storage roots that weigh 50 kilograms or more. And lastly, buttress roots. Certain species of fig and other tropical trees produce huge buttress roots toward the base of the trunk, which provide considerable stability. These are buttress roots. The supporting structure of a vascular plant's shoot system is the mass of stems that extend from the root system below ground into the air, often reaching great height. Rigid stems capable of rising upward against gravity are an ancient adaptation that allowed plants to move into terrestrial ecosystems. Like roots, stems contain three types of plant tissues. Stems also undergo growth from cell division in apical and lateral meristems. The stem may be thought of as an axis from which other stems or organs grow. The shoot apical meristems are capable of producing these new stems and organs. The shoot apical meristem produces stem tissue and intermittently produces primordia that are capable into developing into other leaves, other shoots, or even flowers. The pattern of leaf arrangement about the stem is called phyllotaxy. This probably functions to optimize exposure to the sun for photosynthesis. Leaves may be arranged in a spiral around the stem, or they may be in pairs opposite or alternate to one another, and I'll call that leaf arrangement. The spiral arrangement is common, and for reasons that remain unclear, sequential leaves tend to be placed 137.5 degrees apart. This angle relates to the golden ratio, which is also seen in the angle of coiling in shells of some gastropods. Gastropoda. Humans have used the golden ratio in many ways, including in classical architecture, such as the wall dimensions of the Greek Parthenon, which are based on the golden ratio. The point of leaf attachment to the stem is called a node. The portion of the stem between two nodes is called an internode. A leaf usually has a flattened blade and sometimes a petiole or stalk. The angle between a leaf's petiole or blade and the stem is called an axial, or that leaf axial, and an axillary bud is produced in each axial. This bud is a product of the primary shoot apical meristem, and it itself a shoot apical meristem. Axillary buds frequently develop into branches with leaves or may form flowers. And you should look for the axillary bud, because where you find the axillary bud, that tells you whether that leaf is a simple leaf or a compound leaf, which we'll get to. Neither monocots nor herbaceous eudicot stems produce a cork cambium. The stems in these plants are usually green and photosynthetic. With at least the outer cells of the cortex containing chloroplasts. Herbaceous stems commonly have stomata, and they may have trichomes. Woody stems can persist over a number of years and develop distinctive markings, including scars from buds and leaves that are no longer attached. Bud scale scars. Terminal buds usually extend the length of the shoot during the growing season. Some buds, such as those of geraniums, are unprotected, but most buds of woody plants have protective winter bud scales that drop off, leaving tiny bud scale scars as the buds expand. Some twigs have tiny scars of a different origin. A pair of butterfly-like appendages called stipules, part of the leaf, develop at the base of some leaves. The stipules can fall off and leave stipule scars, which I don't typically discuss. When the leaves of deciduous trees drop in the fall, they leave leaf scars with tiny bundle scars marking where the vascular connections were. And these bundle scars are pretty amazing to see, which you may not be able to see too well here, other than on that right-hand side with figure 35 to 22B as in boy. The shapes, sizes, and other features of leaf scars can be distinctive enough to identify deciduous plants in winter when they lack leaves. And there is a lab for that too. A major distinguishing feature between monocot and eudicot stems is the organization of the vascular tissue system. Most monocot and vascular bundles are scattered throughout the ground tissue system, whereas eudicot vascular tissue is arranged in a ring with internal ground tissue, the pith, and external ground tissue, the cortex. The arrangement of vascular tissue is directly related to the ability of the stem to undergo secondary growth. In eudicots, a vascular cambium may develop between the primary xylem and primary phloem, which I will show you in just a moment. There we go. And I'll show you another figure because this is not my figure reference. This is my figure reference here. In many ways, this is a connected the dots game with the vascular cambium. Connect the rings of the primary vascular tissues. There is no logical way to connect primary monocot vascular tissue that would allow a uniform increase in girth. Monocots do not have secondary growth due to the lack of the vascular cambium. Rings in the stump of a tree reveal annual patterns of vascular cambium growth. Cell size varies depending upon conditions. And let's go show the figure reference there. There is that figure reference, that tree stump. Large cells form under favorable conditions such as abundant rainfalls. Rings of smaller cells mark the seasons where growth is limited. In woody eudicots and gymnosperms, a second cambium, the cork cambium, arises in the outer cortex, occasionally in the epidermis or phloem. It produces box-like cork cells to the outside and also may produce parenchyma-like phyloderm cells to the inside. This is something to very well know and know for your lab tests. Let's continue on. Although most stems grow upright, some have modifications that serve special purposes, including natural vegetative propagation. In fact, the widespread artificial vegetative propagation of plants, both commercial and private, frequently involves cutting modified stems into segments which are then planted, producing new plants. As you become acquainted with the following modified stems, keep in mind that stems have leaves at nodes and internodes between the nodes, and buds in the axials of leaves, whereas roots have no leaves, nodes, or axillary buds. And I see something that we have not gotten to quite yet. Bingo. Let me go back for just a moment. The cork cambium, the cork, and the phyloderm are collectively referred to as the periderm. Cork tissues, the cells of which become impregnated with a water-repellent suberin shortly after they are formed, and which then die, constitute the outer bark. The cork tissue cuts off water and food to the epidermis, which dies and sloughs off. In young stems, gas exchange between stem tissues and the air takes place through stomata. But as the cork cambium produces cork, and this is where I am with the cork, it produces patches of unsuberized cells beneath the stomata. These unsuberized cells, which permit gas exchange to continue, are called lenticels. So what I'm getting at is that lenticels allow gas exchange to occur through the periderm, or through the, let's just say, secondary growth. And this is in woody plants. Now on to modified stems. Bulbs. Onions, lilies, and tulips have swollen underground stems that are really large buds with adventitious roots at the base. Most of a bulb consists of fleshy leaves attached to a small knob-like stem, and for most bulbs, next year's foliage comes from the tip of the shoot apex, protected by storage leaves from the previous year. Corms. Crocuses, gladioluses, and other popular garden plants produce corms that superficially resemble bulbs. Cutting a corm in half, however, reveals no fleshy leaves. Instead, almost all of corm consists of stem, with a few papery brown non-functional leaves on the outside, and adventitious roots below. No picture there. Rhizomes. Perennial grasses, ferns, bearded iris, and many other plants produce rhizomes, which typically are horizontal stems that grow underground, often close to the surface. Here is a rhizome. Each node has an inconspicuous scale-like leaf with an axillary bud. Much larger photosynthetic leaves may be produced at the rhizome tip. Adventitious roots are produced throughout the length of the rhizome, mainly on the lower surface. Runners and stolons, or stolons. Strawberry plants produce horizontal stems with long internodes that, unlike rhizomes, usually grow along the surface of the ground. Several runners may radiate out from a single plant. Some biologists use the term stolon synonymously with runner. Others reserve the term stolon for a stem with long internodes but no roots that grows underground, as seen in potato plants. A potato itself, however, is another type of modified stem. It is a tuber. Now to the tuber. In potato plants, carbohydrates may accumulate in the tips of rhizomes, which swell, becoming tubers. The rhizomes die after the tubers mature. The eyes of a potato are axillary buds formed in the axials of scale-like leaves. These leaves, which are present when the potato is starting to form, soon drop off. The tiny ridge adjacent to the eye of a mature potato is a leaf scar. Tendrils. Many climbing plants, such as grapes and English ivy, produce modified stems known as tendrils that twine around supports and aid in climbing. Some other tendrils, such as those of peas and pumpkins, are actually modified leaves or leaflets. Cladophyls. Cacti and several other plants produce flattened photosynthetic stems called cladophyls that resemble leaves. In cacti, the spines are modified leaves. Leaves, which are initiated as primordia by the apical meristems, are vital to life as we know it. They are the principal sites of photosynthesis on land, providing the base of the food chain. Leaves expand by cell division and cell enlargement. Like arms and legs in humans, leaves are determinate structures. Their growth stops at maturity. Because leaves are essential to plant growth, features such as their arrangement, form, size, and internal structure are highly significant and can differ greatly. Leaves are an extension of the shoot apical meristem. Experiments in which very young leaf primordia were isolated from fern and coleus plants have shown that when primordia first emerge, they do not commit to being leaves. Leaves fall into two different morphological groups, which may reflect differences in evolutionary origin. A microphyll is a leaf with one vein branching from the vascular cylinder of the stem and not extending the full length of the leaf. Microphylls are mostly small and associated with primarily the phylum Lycophyta, and I'll call those the club mosses, a form of seedless vascular plant. They grow around here. Most plants have leaves called megaphylls, which have several to many veins. Most eudicot leaves have a flattened blade and a slender stalk, the petiole, while grasses and other monocots usually lack a petiole, and the leaf wraps around the stem, forming a sheath. The flattening of the blade reflects a shift from radial symmetry to dorsal ventral symmetry. Leaf flattening increases the photosynthetic surface. Plant biologists are just beginning to understand how this shift occurs. In addition, leaves may have a pair of stipules, which are outgrowths at the base of the petiole. The stipules may be leaf-like or modified as spines, as in the black locust, or glands, and vary considerably in size from microscopic to almost half the size of the leaf blade. Veins consist of both xylem and phloem and are distributed throughout the leaf blades. The main veins are parallel in most monocot leaves. The veins in eudicots, on the other hand, are often form an often intricate network. So I would say that they are netted or even scattered. And this shows at the top a eudicot leaf, and at the bottom a monocot leaf. I wonder why they put the title there like that. That title is confusing. And again, I say it should be a eudicot leaf at top at A, and then a monocot leaf at the bottom that's green. Boy, they could have labeled this better by setting a eudicot and monocot leaf. Leaf blades come in a variety of forms, from oval to deeply lobed to having separate leaflets. In simple leaves, such as those of lilacs or birch trees, the blades are undivided, but simple leaves may have teeth, indentations, or lobes of various sizes, as in the leaves of maples and oaks. So here at A, that is a simple leaf, or those are all examples of simple leaves. In compound leaves, shown here on the bottom right, such as those of ashes, box elders, and walnuts, the blade is divided into leaflets. The relationship between the development of compound and simple leaves is an open question. The entire surface of a leaf is covered by a transparent epidermis, and most of these epidermal cells have no chloroplasts. As described earlier, the epidermis has a waxy cuticle, and different types of glands and trichomes may be present. Also, the lower epidermis, and occasionally the upper epidermis, of most leaves contain numerous slit-like or mouth-shaped stomata flanked by guard cells. This shows before you 2 guard cells and then the stoma in the center. And I still can't show you subsidiary cells because these don't have those. At least they don't have those in this cartoon picture. They do exist. The tissue between the upper and lower epidermis is called mesophyll. Mesophyll is interspersed with veins of various sizes. Most eudicote leaves have 2 distinct types of mesophyll. Closest to the upper epidermis are 1 to several, usually 2 rows, tightly packed, barrel-shaped to cylindrical chlororenchyma cells, which is parenchyma with chloroplasts, that constitute the palisade mesophyll. Some plants, including species of eucalyptus, have leaves that hang down rather than extend horizontally. They have palisade mesophyll on both sides of the leaf. Nearly all eudicote leaves have loosely arranged spongy mesophyll between the palisade mesophyll and the lower epidermis, with many air spaces throughout the tissue. And this is showing just that. The upper epidermis, which is a single layer of undifferentiated cells that are transparent with a waxy cuticle at the top. Then just deep to that would be the palisade mesophyll, and then even deeper to that is the spongy mesophyll. And then at the bottom is the lower epidermis and stomata. The interconnected intercellular spaces, along with the stomata, function in gas exchange and the passage of water vapor from the leaves. The mesophyll of monocote leaves often is not differentiated into palisade and spongy layers, and there is often little distinction between the upper and lower epidermis. Instead, cells surrounding the vascular tissue are distinctive and are the site of carbon fixation. This anatomical difference often correlates with a modified photosynthetic pathway, C4 photosynthesis, that maximizes the amount of CO2 relative to oxygen to reduce energy loss through photorespiration. The anatomy of a leaf directly relates to its need to balance water loss, gas exchange, and transport of photosynthetic products to the rest of the plant. And let's now end the chapter with modified leaves. As plants colonize a wide variety of environments, from deserts to lakes to tropical rainforests, plant organ modifications arose that would adapt the plants to their specific habitats. Leaves, in particular, have evolved some remarkable adaptations. Floral leaves, I'll call them bracts. Poinsettias and dogwoods have relatively inconspicuous, small, greenish-yellow flowers. Have you heard of the inflorescence? However, both plants produce large modified leaves called bracts, mostly colored red in poinsettias and white or pink in dogwoods. These bracts surround the true flowers and perform the same function as showy petals. In other plants, however, bracts can be quite small and inconspicuous. Spines. The leaves of many cacti and other plants are modified as spines. In cacti, sharp spines may deter predators. Spines should not be confused with thorns, such as those on the honey locust, which are modified stems, or with the prickles on raspberries, which are simply outgrowths from the epidermis or the cortex just beneath it. Reproductive leaves. Some plants produce tiny but complete plantlets along their margins. Each plantlet, when separated from the leaf, is capable of growing independently into a full-sized plant. The walking fern produces new plantlets at the tips of its fronds. Although many species can regenerate a whole plant from isolated plant tissue, this in vivo regeneration is found among just a few species. Window leaves. Several genera of plants growing in arid regions produce succulent cone-shaped leaves with transparent tips. The leaves often become mostly buried in sand blown by the wind, but the transparent tips, which have a thick epidermis and cuticle, admit light to the hollow interiors, and this strategy allows photosynthesis to take place beneath the surface of the ground. Shade leaves. Leaves produced in the shade where they receive little sunlight tend to be larger in surface area, but thinner with less mesophyll than leaves on the same tree, receiving more direct light. This plasticity in development is remarkable. Environmental signals can have a major effect on development. Insectivorous leaves. Almost 200 species of flowering plants are known to have leaves that trap insects. Some plants digest the insects' soft parts. This reminds me of the pitcher plants, which I'll get to in a moment. Plants with insectivorous leaves often grow in acidic swamps that are deficient in needed nutrients or contain nutrients in forms not readily available to the plants. This inhibits the plants' capacities to maintain metabolic processes needed for their growth and reproduction. Their needs are met, however, by supplementary absorption of nutrients from the insects they capture. Pitcher plants, for example, from the genus Sarracenia, that do live and occur around here in Baldwin County, Alabama, have cone-shaped leaves in which rainwater can accumulate. The insides of the leaves are very smooth, but stiff, downward-pointing hairs line the rim. An insect falling into such a leaf finds it very difficult to escape and eventually dies. The leaf absorbs the nutrients released when bacteria and often the plant's own digestive enzymes decompose the insect's bodies. Other plants, such as sundews, from the genus Drosera, they also occur around here, have glands that secrete sticky mucilage that traps insects, which then digest it by enzymes. And yes, they grow here in Baldwin County. The Venus flytrap produces leaves that look hinged at the midrib. And I think you all have heard of the Venus flytrap. When tiny trigger hairs on the leaf blade are stimulated by a moving insect, the two halves snap shut. This has been your